Our next presentation is Engage Bio, and I'd like to welcome Will Olson, CEO, to the stage.
Hello, everyone. I'm Will, co-founder and CEO of Engage Bio. At Engage, we're developing the Tethosome platform. Tethosomes are non-viral DNA medicines which are potent, durable, non-immunogenic, redosable, titratable, and rapidly scalable. We believe at Engage that there is a vacuum at the intersection of redosability and durability in the current generation of genetic medicines. As we all know, viral therapies like AAV can, in many cases, produce durable expression, but immunogenicity precludes redosing. Further, cost and complexity limit the ability to scale these medicines, in some cases to hundreds of patients, let alone the many patients that one would hope future generations of genetic medicine could treat. We have been very excited about the progress of non-viral medicines like mRNA, which have not only had great success in vaccine and infectious disease applications, but increasingly in redosable therapeutic medicines. But mRNA suffers from a few hurdles for wide adoption across many applications, including brief activity, so mRNA is expressed for some 1 to 3 days, and limited specificity for cell and tissue-specific expression, which limits applications where off-target expression could be deleterious. We are very excited about the potential of non-viral DNA, which we think really checks all of the key boxes: redosability, durability, programmability, and scalability. But non-viral DNA has been held back by 2 key limitations, namely potency and toxicity. And ultimately, non-viral DNA, despite these many advantages, has had very little progress compared to viral and mRNA-based platforms. So to highlight the first challenge, potency is really a function of nuclear localization. So here we have a diagram of a lipid nanoparticle encapsulating a non-viral DNA. When that DNA is introduced into the cytoplasm, by many accounts, only some 1/10,000th of those molecules will ultimately, in a stochastic fashion, end up in the nucleus, which is obviously a prerequisite for expression. On the safety front, we actually think that this is the underappreciated challenge with non-viral DNA medicines. Innate immune sensors in the cytoplasm detect cytoplasmic DNA as a foreign or pathogenic infection, or as a function of a breakdown of the nuclear membrane. And this sets off a whole set of cellular alarm bells and ultimately a cascade of cytokines and pro-inflammatory cell death. If this occurs at a high enough level in an organism, this can lead to lethal immune responses. We've designed the Tethosome platform to solve these 2 key problems. Tethosomes unlock safe and potent non-viral DNA. Here we have, again, the same lipid nanoparticle and the same non-viral DNA, but we've added an mRNA, and that mRNA encodes for our proprietary Tethosome protein. When this mRNA is introduced into the cytoplasm, it's translated, and that Tethosome protein physically shepherds the DNA into the nucleus of cells, thereby increasing the nuclear localized fraction by some 100 to 500-fold. Importantly, these complexes of non-viral DNA and Tethosome protein are invisible to innate immune sensors, rendering our Tethosome proteins invisible to the innate system and ultimately unlocking safe non-viral genetic medicine. Here we show the deleterious effects of DNA in the cytoplasm. We've taken a lipid nanoparticle and encapsulated DNA, Tethosomes, or mRNA, and then compared these to a PBS control. We've administered them at a moderate to high dose, 1 milligram per kilogram of nucleic acids, intravenously into a mouse, and drawn blood at 5 hours. As you can see, interferon alpha and IL-6, which are hallmarks of pathogen infection or cytoplasmic DNA exposure, are very elevated in the DNA condition. This is recapitulated in weight loss data at day 1 and day 2, where there's dramatic weight loss, which is also accompanied by a phenotype of lethargy and ultimately mortality in some animals. With tethosomes, this is completely abrogated, so we see no elevation of interferon alpha or IL-6, no weight loss, and no mortality. So this means that we can provide the same coding information as traditional DNA, but without triggering immune reactions. On the potency front, here we have a model where we've taken a firefly luciferase and encoded it in tethosomes, DNA, or mRNA, respectively. We've then used bioluminescence imaging serially over about 22 weeks to monitor the level of expression from the liver of these animals. As expected, mRNA produces very high levels of expression on day 1, but this rapidly falls off over just a few days. DNA, and in this case, this is an optimized DNA, codon optimized, CpG depleted, promoter optimized, so it's quite good. It does lead to durable expression, but ultimately, the toxicity that I noted a moment ago and the very low level of expression, some 3 logs lower than mRNA, is likely subtherapeutic for most use cases. With tethosomes, we think we have the best of both worlds. So we do achieve this very high level of protein expression, but it's durable, in this case, with a very flat PK curve over a matter of months. And with this level of durability and no relevant limits on cargo capacity, we think this opens the door to many potential use cases for tethosomes. We like to make a distinction between our approach and so-called one-shot genetic medicines. So unlike AAV or other viral therapies, our non-viral system is redosable and titratable. That means that we can come in with a very low starting dose, here 0.1 milligrams per kilogram, and then titrate up to the optimal level of expression. This allows us to avoid 2 challenges with current viral genetic medicines. One is undershooting the target level of expression, where patients don't receive ultimately a therapeutic level and then they cannot redose. But the other, which is emerging in the case of some of the first generation factor VIII therapies, is supraphysiological levels of expression, where if a patient has 2 to 3 times the normal level of factor VIII, ultimately that patient may need to take anticoagulation medications for an indefinite period of time. So we think that this titratability and redosability unlocks optimal expression levels. Now, with a new class of genetic medicines, there are many potential use cases that we can apply this to. I'll highlight 3: gene replacement, expression of any therapeutic protein, not necessarily associated with a monogenic disease, and then oncology applications. Within gene replacement, we've chosen factor VIII as our proof of concept molecule because there's so much data within viral and other non-viral platforms against which we can benchmark ourselves. Again, with the ability to titrate and redose, patients won't be afraid to waste their one shot, as actually is the case with the first generation AAV factor VIII products. Here we show an experiment where we've taken human factor VIII and encoded it in our tethosome platform, and we've administered a single dose to mice. We're then measuring the level of factor VIII in plasma, and as you can see, we've achieved 50 to 100% of normal levels with a single dose. So we think that with the features of our platform, this could potentially overcome the significant burdens to adoption that have been faced by first-generation AAV products in this space. Within protein therapeutics, we've picked what is a very hot target at the moment as our proof of concept. This is a GLP-1 receptor agonist. We've encoded this in our tethosome system and again administered a single dose to a diet-induced obesity model. We're seeing right around the exact same levels of weight loss that one would expect with subcutaneous weekly dosing in humans or in many mouse models with a much more frequent dosing regimen. Because we have such a significant cargo capacity and flexibility within our constructs, this means that we can potentially encode multiple transgenes, such as so-called muscle-sparing peptides, which actually allow for retention of muscle mass as all of this fat mass is lost. So we're very excited about the potential for combination therapies in this space. Finally, within oncology, we're most excited about the potential for our platform to achieve tumor-specific expression. So because we're employing DNA, we're able to use DNA logic circuits that only activate in cells which also express a target cancer biomarker of our choice. So this can be thought of as the following logical statement. If a tumor expresses transcription factor X, in this case we would pick a transcription factor that is not expressed in healthy adult somatic tissues, but is greatly overexpressed in cancer, then and only then our payload will be expressed. So this allows us to use systemic dosing of vehicles that have promiscuity, so they'll go to many different tissues and potentially multiple tumor nodules, but leading to expression only within the tumor. The level of specificity of our leading constructs really does approach approximately 100%. So here we have a model where we have taken a human xenograft cell line and engrafted it in the lower left flank of the mice. We've then administered our tethosome, mRNA, or DNA, each encoding, again, a firefly luciferase. As you can see, mRNA does lead to expression in the tumor, but the vast majority of the expression is originating from the liver in the upper abdomen as a function of the biodistribution of these particles. This is true even when one employs the tricks like microRNA binding sites and RNA binding protein sites, which can confer a degree of tumor specificity. Ultimately, the level of regulation that's available at the mRNA level is insufficient to drive truly perfect tumor specificity. With DNA, as you might be able to see, there's a single red pixel in the tumor, so we are able to confer tumor-specific expression, but ultimately this is likely subtherapeutic levels of expression for almost any transgene. But with our tethosome system, we achieve the very high levels of expression that one might see with mRNA, but without any expression in off-target tissues such as the liver. This is also shown on the right, where we've excised tumor, liver, and spleen, and shown that indeed, mRNA does lead to expression in each of these organs, but really with the tethosome system, we're able to turn off completely expression in the liver and spleen. So we've taken as our proof of concept molecule in oncology a hepatocellular carcinoma model. We like HCC for a number of reasons. One is it's relatively transfectable compared to other solid tumor targets. And also there are great tumor-specific promoters that we can employ. In this model, we're only activating the innate immune system. So we have a basket of cytokines, which are pro-inflammatory, and even just engaging the innate immune system, we're able to achieve significant tumor growth inhibition and significant increases in survival. In this ongoing study, we have not yet reached median survival in the treatment arm. So we have a lean and nimble team, and just in the past 2 years, we've built out this platform and demonstrated it in a number of in vivo applications. Early next year, we hope to nominate our first candidate and begin large animal toxicity studies. But we recognize that we can only do so much as a small team. And so we're here to connect with potential pharma clinical development partners in order to not only accelerate our progress on the programs that we've illustrated, but really to broaden the potential reach and number of applications that we can pursue. So again, I'm Will Olson, CEO and co-founder of Engage Bio. And if you're interested in learning more about our tethosome platform, I would be happy to connect after the talk. Thank you all.